Thank you very much, Nick, for the kind introduction. Actually, I'm going to be much more forward-looking than that today. As you well know, vaccines have had a substantial impact on human health, but we have many formidable challenges ahead of us, not the least of which is improving existing but suboptimal vaccines. And as Nick nicely summarized, influenza is a vaccine that's widely used but is very marginally effective, both from a potency and efficacy perspective, but also the way that we actually manufacture it. Secondly, we have a lot of unmet medical needs, very large unmet medical needs that could conceivably be addressed by a vaccine, such as HIV. And so the focus of my talk today is going to be more about how we could facilitate a rapid response, and it's certainly timely right now. We're grappling with a situation where we have an infectious disease that's spiraling out of control, and it would be very nice to have a vaccine, but we haven't really spent a lot of time over the many decades since Ebola was first discovered. Today we could just very easily have a vaccine in our hands, but we just didn't take the trouble or the effort to actually make one. And one of the reasons is we don't have a very effective way to have a rapid response to newly emerging or re-emerging infections. Let's see if we can move this. There we go. In order to have advancements in any of these three areas, though, we're going to need to identify and apply new technologies. And these could come in the form of paradigm shifts, such as comprehensive ways to identify the protective antigens out of a pathogen, or the use of the power of synthetic biology to better understand the structures that we need to present to the immune system to have effective immune responses. I've alluded to synthetic vaccinology, and this is not only a practical issue of getting away from using biological systems altogether to manufacture vaccines, which could certainly streamline the development and perhaps reduce the cost and the time to get to a product, but also it's probably the only way that we're going to really have a truly rapid response to a newly emerging infection. And then finally, if we can better understand how the host responds to the pathogen and the vaccine for that matter, we can start to take rational approaches to designing the vaccines to be more effective. And we don't have to look much back further, and you saw this graph already this morning once, to see how woefully inadequate we are in terms of a rapid response. As Nick mentioned, by the time we had sufficient vaccine in the stockpile, the disease basically had come and gone. And there are a number of things that we can do upstream of this to give at least an incremental increase in the speed with which we respond to an influenza outbreak. But what we really need is a fundamentally different way to manufacture the vaccine. And the technology that I'm going to speak about today is a nucleic acid-based technology based on RNA that's actually been the subject of several decades of research, both in our organization and others, to use a viral vector system based on an engineered alpha virus genome. And up till recently, we have been focusing on delivering this RNA genome in the form of a viral particle, like a viral vector. But we, about 5 or 6 years ago, decided that we needed to get away from using biological systems for a variety of reasons. One, just the practical issues of making biologics, but also the potential safety issues, and the issue that plagues viral vectors in general, and that is when you use them multiple times, you engender immunity against the vector itself, and thereby limiting its utility in the long run. The approaches we're taking are twofold, using lipid nanoparticles to encapsulate the RNA, or emulsions that have been rendered cationic on the surface, to which you adsorb the RNA onto the surface. To give you a little idea of what the genome looks like, it's, as I said, based on an alpha virus genome. It contains the non-structural proteins which encode the replicase machinery that allows the RNA to replicate in the cell. But we've changed the downstream genes and replaced the structural genes with the gene of interest, such that when the RNA gets into a cell, either through direct fusion of the membrane or by facilitated entry in some sort of phagocytic pathway, once this RNA is in the cytoplasm of a cell, the expression of the non-structural proteins allows the RNA to make many copies of itself, and it's calculated that a single copy of RNA can make tens of thousands of copies of itself. There's an internal promoter that drives the expression of the mRNAs encoding the genes of interest or the structural genes in the wild-type virus, and this gives you the amplification effect of a small number of incoming RNA molecules can give many thousands of copies of your gene of interest. Another reason why this technology might be particularly effective is that the double-stranded RNA intermediates in this process are known to interact with the innate immune system, so you have a built-in adjuvant effect. So from a rapid response perspective, there are 4 key attributes that one would want to have. The first is, since you may not know exactly the type of immunity that you need to counter the pathogen, you may want to have a technology that induces broad and effective responses, both antibody and T-cell mediated. And we've done a number of preclinical studies with this technology to evaluate, one, not only how potent it is, but what's the quality of the immune responses that it induces. Here on the left, it shows that these delivery systems work at least in part by protecting the RNA from degradation. This is an unformulated RNA that has been put in a test tube with RNase, and you can imagine that naked RNA is very rapidly degraded by this enzyme. But if you formulate it either with the LNP or the CNE, you get substantial protection of the RNA from degradation, which probably gives it the time it needs to get inside a cell before it's degraded by the tissue nucleases. On the right here are some mouse data to show that, in fact, this formulation is effective. Here's a 1 microgram of RNA dose given as naked RNA, and you can see that, surprisingly, even naked RNA can get into a cell, express the antigen, and induce an immune response, as measured here by antibodies against the RSVF protein. But if you formulate that RNA, in this case with the LNP, even at a 10-fold lower dose, you get substantially higher immune responses that are comparable to our benchmarks here, which is the viral replicon particle. It's the same RNA genome, but delivered by the viral vector, and the DNA encoding the same gene delivered by electroporation. It also is very effective at inducing functional immune responses. Here are RSV neutralizing antibodies. Here's the formulated RNA compared to naked RNA, and again, benchmarked with our viral vector. And here is protective immunity in a challenge model in cotton rats. The other interesting feature of this technology, like other nucleic acids, is that you can co-express multiple proteins. And in this case, you can express multiple genes of CMV, which are known to form a complex. And you can get higher levels of potency by co-expressing these genes in the same cell. And presumably, you get a formation of this higher ordered structure, which induces neutralizing antibodies that are common to the structure, but not common to the individual genes. And then finally, from a T-cell perspective, we know that it can induce both potent CD4 and CD8 T cells that are polyfunctional. And again, compared to benchmarks, they favor quite well. You can administer this vaccine by the intramuscular or intradermal or subcutaneous route, although it seems, at least in the animals that we've tested, intramuscular seems to be the favored route for potency of immune responses. It's also effective in larger animals. This is a study with RNA encoding HIV envelope in non-human primates. And again, we're benchmarked it against the viral replicon particle at a dose of 1 × 10⁸, which is the dose that Alphavax had shown to be effective in humans at inducing immune responses against a CMV viral vector. And envelope protein formulated with the oil and water emulsion adjuvant MF59 at a dose of 100 micrograms, which is again a dose which was demonstrated to be potent and immunogenic for induction of neutralizing antibodies in humans. So these are 2 benchmarks that we know are effective in humans for immunogenicity. The middle panel here shows the RNA vaccine encoding HIV envelope at a dose of 50 micrograms, and we've immunized 3 times with the RNA and then boosted twice with protein. And you can see that even in the priming phase, you have very potent levels of antibodies directed against HIV envelope at a dose of 50 micrograms of RNA. When we looked at the functionality of these antibodies, we found that the RNA vaccine, quite surprisingly, gave equivalent levels of neutralization compared to the adjuvanted subunit protein, and were better than the viral replicon particle. And as expected, the T cell responses here, as measured by interferon gamma ELISpots, were superior for the RNA vaccine over the subunit envelope protein with MF59, which wouldn't be expected to induce a Th1 response, but also the viral replicon particle, where we're getting 4 to 5 times higher levels of Th1 immunity in these primates compared to the viral vector form of this replicon particle. The last benchmarking study we did was with 2 licensed products, and this was influenza in the ferret model, which Nick had alluded to, which is the gold standard for using flu vaccines for protective immunity. The 2 vaccines that we've benchmarked against were the H1N1 subunit vaccine, which is in the seasonal vaccine. This is a full human dose of 15 micrograms. This is the vaccine that is given to the general public. We also have an adjuvanted version of this vaccine in MF59, again at a full human dose of 15 micrograms. And this is the vaccine that's administered to the elderly, and it's known that MF59 markedly enhances the potency of this H1N1 vaccine. If you focus on the blue colors first, they're the post-dose 1 titers as measured by virus neutralization here. And you can see that 5 or 15 micrograms of RNA given either once or twice, shown in the red here, give equivalent to or better than immune responses elicited by the unadjuvanted licensed product, and equivalent to the MF59 adjuvanted licensed product, which is quite remarkable. We've never seen any vaccine technology that approaches the MF59 adjuvanted subunit in this model. Interestingly, if you look at protection, the MF59 adjuvanted product is in the black circles, the unadjuvanted down here, and you can see that those vaccines were protective, but certainly not completely protective. The RNA-based vaccines, which are in the lighter yellow and green here, show protective levels that appear to be even superior to the MF59 adjuvanted subunit, probably due to the contribution of T cell responses in these animals that is not afforded by the MF59 adjuvanted subunit H1N1. So this vaccine is potent and induces broad immunity in a wide range of animal models. The second attribute would be efficacy against a variety of different pathogens. And this table summarizes some but not all of the targets that we've tested, both internally as well as during collaborations. And you can see that we've shown immunogenicity and efficacy against a variety of different viral targets, up to and including large animals like cattle and non-human primates. Collaboratively, working with USAMRIID, we've shown that in the guinea pig model, RNA encoding the glycoprotein from Ebola is protective. And collaborating with Yale University, we've demonstrated that RNA encoding a malaria antigen is also immunogenic and protective. So it does appear that this is a technology that has broad utility across a number of different pathogens. The third aspect, more practical aspect, is clearly how quickly can you respond? How fast can you go from knowing what the pathogen is to having a vaccine in your hand? And one of the things that we have going for us is a well-established manufacturing process for making RNA. In vitro transcription of RNA has been around for quite some time, well-established, rapid, and in fact, CureVac, who's working with Sanofi, has already established GMP production of messenger RNA, which, albeit a little smaller than the RNAs we're using, which are in the order of 9 to 10 kilobases, demonstrates that it is possible to make large batches of RNA under GMP conditions. And just on the right here shows if you make RNA with the kit or if you make RNA with our process, you end up with the same levels of productivity. In terms of how quickly you could go from sequence to RNA, this is just a schematic showing the specific steps in making the RNA. As I mentioned, it's produced using an in vitro transcription reaction, and very standard procedures for purifying and filtering and sterilizing the RNA. You can go from starting this to having RNA drug substance in a vial within about 6 to 8 hours. The process gives you very high purity of RNA, approaching 100%. And we've done dozens of different reactions encoding different genes that give yields in the range of 5 to 6 milligrams of RNA per mil of in vitro reaction. So it does appear to be a very generic process that's independent of the sequence of the gene that's in your construct. So we did have the opportunity to test this in real time, and this is our response to the H7N9 outbreak from 2013. And because of an existing ongoing relationship we had with the Craig Venter Institute to make synthetic seeds for manufacturing virus for influenza, we had at our disposal synthetic genes encoding the HA from H7N9. So we obtained those from the Venter Institute, used that as our template for manufacturing the RNA encoding the HA from H7N9. And from the time that the sequences were posted on the internet to the time which we had RNA in a vial ready to immunize was 8 days. And looking back on this, we know that we could shave several days off of this because there are 2 snail mail steps of mailing things between sites which we could eliminate by having everything in one location, and also having certain reagents and processes in place. We think that we could do this in a matter of 4 to 5 days rather than 8 days. This RNA vaccine was used to immunize mice, and within a couple of weeks we had levels of neutralizing antibodies that were quite significant. So conceivably, one could go from sequence to protective immunity in a matter of weeks. The last is having capacity to produce this vaccine at scale. It's one thing to make it fast, but you need to have a lot of doses as well. And so based on the data we've generated with our various generation of RNA vaccines, we can start to make some estimations of how many human doses we might be able to make. The first generation vaccine that we tested in non-human primates with the HIV construct showed that it was effective at a dose of 50 micrograms. If this was the human dose, this would give us, based on our experience, about 80,000 human doses per liter of in vitro transcription reaction in 6 hours. The second generation vaccine that we tested in the ferrets showed that it was effective at 5 micrograms, gave comparable, if not better, immunity compared to licensed products. If this were the human dose, then we're up to 800,000 doses per liter, and we are developing third-generation technologies which so far look to be 5 to 10-fold more potent in animal models than the second generation. So now we're starting to get in the range that if this scales up to humans, we're able to produce millions of doses of vaccine in a matter of hours from 1 liter of in vitro transcription reaction, which is certainly a scalable manufacturing process. So one could envision that a truly rapid response could be something like this, where you have local sentinel sampling to identify newly emerging or re-emerging pathogens. As soon as those pathogens are identified, the sequence is determined, they could be put on the internet in a cloud, and this information transmitted to local manufacturing sites, which then could very rapidly produce your vaccine at scale and have vaccine that could be used to stem an outbreak. So in summary, synthetic vaccinology, I think, has 2 real tangible benefits. One, the practical aspect of streamlining, lowering the cost, and speeding up the development of vaccines by getting away from biological systems altogether. And if you couple this with bioinformatics and digital information technology, you could envision a system where you could very rapidly respond to a newly emerging infection. One of the technologies that has the potential to address this is the self-amplifying RNA-based vaccines that I described. They have the potential to combine the positive attributes of many other types of vaccines, live, attenuated, whole organism, viral vectors, DNA, for example, without some of the inherent limitations of those vaccines. They are able to, at least in animal models so far, induce broad and potent immune responses. The processes for manufacturing appear to be generic, so that you could have a plug-and-play type platform technology, and, as I've mentioned several times, would be one way to have a truly rapid response to a newly emerging infection. Thank you very much for your attention. I'd be happy to answer questions.
So, Jeffrey, very exciting technology. Can you tell or give us an indication of when do you think this will sort of be approved for human use, and also do you have an idea of what will be your initial antigen that you would take into phase 1? Like, do you have a candidate in mind at the moment?
Well, our RNA is still preclinical at this stage, so it's almost impossible to predict when such a vaccine would be licensed. But CureVac, which is developing a messenger RNA-based vaccine, is already in phase 2 with their product. So they're approaching licensure with their vaccine. So I think the technology has been validated already in humans, both from the perspective of the viral particle-based delivery of the replicon by AlphaVax, which showed that that particular type of RNA is immunogenic, and messenger RNA being immunogenic in several models so far, both cancer as well as influenza. From CureVac, I think the path is not so risky, so I think there's a reasonable chance of success with this technology. As far as targets, I think obviously viral targets would be logical, but as you've seen, the technology appears to be effective not only for viruses but also for parasites like malaria, and I didn't have on my table, but we also have evidence that the RNA vaccine is effective against bacterial targets as well. So I think the world is open in terms of which targets one could envision for the technology.
Okay, so
Jeffrey, quick question. You know adeno and all those things, parenteral injection, and then become tolerant. And with your so-called LNP RNA vaccine, you have that problem too. You cannot boost. You only prime one time. That's it.
There should be no anti-vector immunity with this, since there's no proteinaceous material in the vector itself. It's based on lipids or emulsions and RNA. So there's no protein, unlike in a viral particle where you can induce potent anti-vector immunity. That isn't to say that there may be interfering T-cell mediated anti-vector immunity targeted toward the non-structural genes of the vector. We haven't seen that yet. But it's certainly something we need to look at. But I think there's much less likelihood of interfering anti-vector immunity with this than conventional viral vectors.
I'm wondering, as much as the efficacy of the vaccine is demanded, the reactogenicity should also be no issue. So I'm wondering, in this self-amplification process, how will you control for the pro-inflammatory responses, in particular if you affect also the innate immune system? It might be very difficult to control this, and you might end up with reactions you don't want to have, even though the vaccine is very immunogenicity.
You bring up an excellent point. It's not just from a practical perspective of reactogenicity, but we know that the replicon is very sensitive to interferons. And so if you overstimulate the interferon response on the way in, which is a natural antiviral response of host cells, you can prevent launch of the replicon. So we need to strike a balance between having the built-in adjuvant effect of the double-stranded RNA, but at the same time not overstimulating before you've allowed the replicon to launch. And so we've been playing around a little bit with ways to manipulate that, so far not with a lot of success. But from a reactogenicity perspective, the RNA component of the vaccine is very well tolerated and safe. I think that the formulation has more likelihood of being reactogenic than the RNA portion.
So Jean-Pierre, and then over here.
I was wondering if you looked at cytotoxic T-cell responses induced with your RNA-based vaccine.
Yes, it's very effective at inducing CD8 responses, in fact, more so than the viral vector system. As far as CD8 responses in primates, as far as we've gone is looking at CD8 ELISpot responses. So we haven't looked at functional lytic cells in primates, but we know they are very effective at inducing CD8 responses.
Okay. A couple of quickies, Jeff. How long does the immune response last for? And can it be... Question two is, there's a bit of a sort of an oxymoron to some extent. If you want the vaccine to be available rapidly, that's one thing, that's great. On the other hand, if you have a cross-protective vaccine of long-lasting immunity, why do you need a rapid response to a vaccine anyway? Because you're protected.
Well, to answer your second question first, I mean, That may be true for flu if we can come up with ways to better induce long-lived cross-protective immunity, then it would obviate the need for this, but that doesn't solve all the other newly emerging pathogens like MERS and SARS and Ebola. And your first question, in terms of longevity, in small animals, it's like most vaccines, it lasts a long time. In primates, the immunity does wane, as it does with many other kinds of vaccines, but it's boostable. If you recall the primate data with HIV envelope, you could see that it rapidly induced responses during the priming phase, which slowly decayed but could be boosted tenfold by follow-up boosting.